To understand, let's assume we are having a test tube containing our sample. An incident light of intensity I0 is being used here. The sample is absorbing the light and the light of intensity I is coming out. According to Beer-Lambert's law, the log of I0 upon I is equal to absorbance A, which is finally equal to the multiplication of three parameters epsilon, C, and L. First of all, in this equation, I0 is the intensity of incident light and I is the intensity of emitted light. The ratio, the log I0 upon I is equal to absorbance. Now, absorbance is the main parameter of the whole concept. We use it to find out how much light is absorbed by the sample. Now let us talk about other parameters, that is epsilon, C, and L. Here, epsilon is molar extinction coefficient, C is the concentration of sample we are using, and L is the path length. Now, what do we mean by path length? Path length is actually the length by which the light is passing. In this case, this is the length of our test tube or of our cuvette where the sample is present. This length is generally measured in decimeters or centimeters. Now, what do you mean by epsilon or molar extinction coefficient? The molar extinction coefficient is the measurement of how strongly a chemical species can absorb the light. More the light absorption, more the value of epsilon. It is an individual property of the chemical species, which means it depends upon the nature of species, and for different chemicals, the value of epsilon or the value of molar extinction coefficient will be different. Here in this equation, if we just keep the value of concentration and path length is equal to 1, then the value of absorbance will be equal to the value of molar extinction coefficient. So till now, what we have learned? We have learned from Beer-Lambert's law that the absorbance of light by a given sample depends upon the concentration of sample and the path length. And finally, it also depends upon the nature of sample in the form of molar extinction coefficient.